The general structure of viruses is our next section, but before we get there, let's zoom out and see their relative size. So if we look here with our kind of, I don't know, seafoam green yeast cell here, this is going to be the largest one on this graph at roughly 7 micrometers. This one's the largest on this picture because it is the only eukaryote. So eukaryote's going to be the largest cell. Then if we look at our pink and purple ones up here, these are our prokaryotes, and E. coli, streptococcus, and so on. And then we can see from about 1 micrometer is our largest virus all the way down to 22 nanometers with yellow fever virus. This is the relative size of our viruses. And then for perspective, we have the smallest molecule on this picture, the hemoglobin molecule found in your blood. It's a protein molecule, is even smaller at 15 nanometers. Okay, so viral components, we're going to go through capsids, nucleic acids, envelopes, but before we get there, viruses resemble their own thing. They're not cells, they're not even trying to look like cells. And some key things to keep in mind are they have no protein synthesizing machinery. So I know chapter 8, we haven't got there yet, but chapter 8 goes through, okay, we go transcription, translation, how do we make RNA? How do we take that RNA and make protein? Viruses still need protein, but they don't have any of their own machinery. Their outer surface will be composed of regular repeating subunits that will give them a more crystalline appearance. So they're not going to have a cell wall and their own fancy cell membrane. Instead, they're going to have a very geometric looking appearance that is simple and compact. Most viruses will contain only the bare necessities, only exactly what they need to invade, get inside their host, make more of themselves, and get coded and sent out to find new hosts to infect. That means they'll have an external coat, their nucleic acids, maybe if they're being extra fancy, 1 or 2 enzymes. Okay, so viral particles, all viruses will have a capsid. So I'd encourage you with this next section to keep in mind keywords like all and some. So all viruses will have a protein shell known as a capsid that surrounds their nucleic acids. Some of them, in addition to that shell, will have an envelope. So on the outside, if we look at the covering of a virus, they're all going to have that very geometric capsid in the pink here, and then some of them will have an envelope on the outside as well. Usually that envelope is stolen from their host cell membrane. So they kind of just use it as an extra like disguise jacket on the outside to evade the host's immune system. Viruses with only the nucleocapsid are known as naked viruses. Okay, so we can have enveloped where they'll have that cell membrane that's stolen on the outside. If they only have the capsid, they're a naked virus. Both naked and enveloped viruses will need something to help them dock with their host cell, and those are known as spikes. So you'll often see a very geometric shape with capsid with little like spiky bits coming off. That would be what they would use to dock to the receptors on the cell membrane of their host. A viron is a fully formed virus that's ready to go and infect their host. Okay, so naked versus enveloped viruses, you'll see both of them very geometric. Both of them have their nucleic acids inside. Both have their capsid of the proteins that are repeating on the outside. And then You'll also notice both have these spikes that they would use to dock with their host. Main difference here is one stops there, the other one includes an additional envelope out here in yellow. Each capsid is constructed from identical subunits known as capsomeres. So if we go back a slide, each one of these little blue spheres here would be a capsomere. Each one of these little triangles, capsomere. They're repeating subunits that are identical. Think of it like you had a whole bunch of red Legos, you put them all together. That would be like the capsomere to, I don't know, your tower you're creating, okay? Capsomeres can self-assemble, so they don't need a special enzyme to put them all in place. They just find the lowest energy state, which happens to be formed into the capsid. Typically, they'll arrange either into a helical or icosahedral structure. So helical kind of being more like tube shaped, or icosahedral, a 20-sided, very geometric, kind of more sphere with sides on it structure. Okay, so we're going to zoom in on each of these. First, we have our helical capsids. Those are the simpler of the two types of structures. Helical capsids are made of rod-shaped capsomeres. So again, we have these rod-shaped capsomeres in blue. All together, they form the capsid around the nucleic acid in purple, kind of squiggling through. The helical capsid will form kind of this like bracelet-like shape, kind of a tube or a rod around the nucleic acid. One example of a naked helical capsid virus would be the tobacco mosaic virus that was discussed at the very beginning. We can also have enveloped helical capsids. So you'll see we have the DNA in purple, and then we have that kind of bracelet helical capsid inside, and then that's further encompassed into an envelope in yellow outside here. An example of an enveloped helical capsid virus would be SARS-CoV-2, rabies, measles. This is a very common type in animal viruses. You can also have icosahedral capsids. So an icosahedron, if you think back to geometry, is a three-dimensional 20-sided figure. So you'll see here, if you counted like 1, 2, 3, they kind of have different faces depending on how you turn it. And they're made up of many different capsomeres, these little blue ones here. Poliovirus has 32 different ones, whereas like an adenovirus, 252 of them, but they're all going together to form a 20-sided shape. You'll also notice they have their spikes out here. If you look at the electron micrograph over here, you can see the icosahedral shape here with the little spikes popping off. Can also have enveloped icosahedral viruses. Same idea, where you have the DNA core with the icosahedral capsid and then a further envelope outside of that. This would include things like hepatitis and herpes virus. The final type is the complex capsid. These are found only in bacteriophages or viruses that can infect bacteria. They'll have multiple types of proteins, and their overall structure won't be symmetrical. So you'll still see they will still have a nucleic acid. They'll still have a capsid, but then they have these additional proteins to help them attach to their host. So like tail fibers, tail pins to help them kind of attach, hold on to, and insert their genetic material. Every time I see these, I always think this looks like the start to, I don't know, a black and white old movie with aliens, and it's like the alien spaceship. I don't know, but these are the most bizarre looking capsids. Okay, so another thing we can use to further identify and characterize viruses is their nucleic acids. They have a genome of their own, even though they're not technically alive here, they still carry their own unique genetic material. Unlike cells, though, that have both DNA and RNA, so if you think of a eukaryotic cell, they'll have their DNA in the nucleus, and then they'll go through transcription and make copies of the genes they want to use and make RNA. And they'll have both. Viruses, though, have to choose. They have either DNA or RNA, but they are not going to have both. The viral genomes are very small compared to those of a living cell. So to give you some perspective, viral genomes are usually between 4 to 200 genes, which you say, go, hey, 200 genes, that's pretty good. Yeah, well, your genome is around 23,000 genes, okay? So much, much simpler, much more concise. Their genomes can be double-stranded DNA. So we think of like the DNA helix with the base pairs binding to each other, forming that kind of ladder shape, or they can be single-stranded, only half of that ladder. The double-stranded DNA can be linear, so like our chromosomes, or they can be circular, more like that of a prokaryote. We can also have double-stranded RNA, so still having A's, T's, and U's, uracils, so the same base pairs you would find in RNA, but they could be double-stranded, looking more like DNA, or they could be single-stranded. Single-stranded RNA genomes can be positive sense in that they're ready immediately when they get into the host to be made into protein, or negative sense where they need to be converted and processed more fully. Singular nucleic acids or segmented ones are also options. So an RNA genome could be one piece of RNA that has all the genes, or it could be segmented, multiple pieces of RNA all inside of that capsid. Retroviruses are viruses that carry their own enzymes. Okay, so now they're not going to have a lot of them, but maybe one, maybe two of their own enzymes that can be used to convert RNA into DNA. Okay, so zooming in a little bit more on each of those, you have double-stranded DNA, which looks a lot like the DNA helix we all know and love in purple here. Example would be like smallpox or herpes, okay? You can have single-stranded DNA, which only is half of that. You can have single-stranded RNA, that's the positive sense, it's all ready to go immediately when it gets into the host, like polio. Single-stranded negative sense, that would be like influenza, where it has to be further processed before it can be used. You can have double-stranded RNA or single-stranded RNA, and in this case, the virus would also have enzymes to convert it to DNA. These enzymes are known as reverse transcriptase, so they can take the RNA molecule and read it backwards basically and convert it to DNA. So to reiterate here, viruses will always have the protein capsid. So they're always going to have those capsomeres that are repeating, forming a helical structure or an icosahedral structure around their nucleic acids. Sometimes they will have more proteins and lipids in an envelope. So we can have naked viruses or we can have enveloped viruses, but that's sometimes. All of them should have nucleic acids. It could be DNA, it could be RNA, it could be single-stranded, it could be double-stranded, but they have to have some form of genetic code. Sometimes they'll have enzymes. So they could have a polymerase, an enzyme that can synthesize DNA or RNA, or maybe they'll have an enzyme like HIV has, where they can convert RNA into DNA with a reverse transcriptase. Most viruses, though, lack the genes for synthesizing metabolic enzymes. Instead, it's just more efficient for them to rely on their hosts and hijack their machinery.